Okay, so soil disturbance is an important consideration at seeding time, particularly with tine systems which are known for higher disturbance relative to discs. Soil disturbance can affect the soil moisture loss and weed seed simulation at seeding. Lateral soil throw is a particularly important parameter of soil disturbance, and it can reduce the amount of furrow backfill as well as cause interactions with the adjacent seed rows. These interactions can throw soil onto the adjacent furrow, increasing seeding depth and compromising seeding depth accuracy. It can also throw herbicide onto the adjacent row, compromising crop safety. Here's an example of furrow ridging, which is where the soil is thrown onto the adjacent rows. The picture on your right shows the middle row, which has suffered trifluralin damage from herbicide being thrown from the adjacent rows. So how does soil throw affect farmers? To limit ridging, row spacings have to be limited. This means that farmers have to go for wider row spacings and can't go narrower to increase competition with weeds. Also, as soil throw is a function of speed, it typically limits the operational speed of tine systems and thus the work rate and timeliness of sowing. So to help understand soil movement, Dr. Ali Akbar Soldier of UniSA conducted a study using soil traces to track how the soil moved. He used clod-sized PVC soil traces in a remoulded soil bin. He studied the effect of rake angle and different blade face geometries, which are discussed briefly in the paper, but I don't have time to talk about them now. He also looked at a Bentleg furrow opener. The Bentleg furrow opener offsets the foot from the shank, the idea being that the foot upheaves the soil and the bend offsets the shank from the main upheaval, limiting its interaction with the loose soil. Here's some furrow profiles from his work. As you can see, the Bentleg has a lot less soil layer mixing, with all the colours being in the same row. It also just tends to lift the entire soil profile, rather than pick it up and throw it sideways, creating furrow, emptying the furrow here. So the work by Dr. Ali Akbar was great, and it gave us an insight into how soil moves. But it was limited in a couple of ways. It was carried out in an indoor soil bin, in a remoulded soil with no organic matter or root mass, and it was only carried out at one speed of 8 kilometres an hour. So in September last year, a field trial was conducted out at Roseworthy, looking at these 4 openers you can see here to validate and extend on the work of Ali Akbar. The two openers had straight shank, one with a 90 degree and one with a 53 degree rake angle, and the 2 Bentleg openers had varying degrees of offset, one with a 45mm and one with a 95mm offset. They were tested at 3 different speeds, 8, 12 and 16 kilometres an hour, in a dry soil condition to simulate dry sowing. Both force and furrow parameter data was collected. The force was collected using a 3-dimensional force frame with load cells in the vertical, side and draft directions, and a laser profile scanner was used to collect furrow parameters. This was done by taking a laser scan of the soil surface, and then the loose soil was removed and the furrow profile was scanned. Here's the lateral soil throw results from the trial. As you can see, both Bentleg openers were able to maintain low soil throw at 8 kilometres an hour relative to the straight shanks. However, the Bentleg 45mm showed a sensitivity to speed as the soil throw reached similar levels to the straight shank openers. The Bentleg 95 with the large offset was able to maintain low soil throw at speeds up to 16 k's an hour. It was a similar story for the furrow backfill, or soil that flows back around into the furrow. The Bentleg with the large offset was able to maintain 100% furrow backfill, while the Bentleg with the small offset and the 2 straight shanks tended to empty the furrow as speed increased. The straight shank opener with the low rake angle at 53 degrees tended to empty the furrow the most. To help understand these 2 results, here's the furrow profiles from the straight shank with the 53 degree rake angle and the Bentleg opener with the large offset on the bottom. As you can see, the straight shank has emptied the furrow creating a dip area here and throwing soil laterally here with a large spillover from the furrow boundary. The Bentleg opener, however, tended to lift the soil profile and not spill much soil past the edge of the furrow boundary, only spilling around 50 mils compared to around 250 mils with the straight shank. The draft force results for the openers were as expected. The lower rake angle was able to had lower draft requirements. The Bentleg openers were able to maintain low draft requirements due to a 45-degree rake angle on the leading foot. All the openers' draft increased by around 10% each 4 kilometre increase in speed. So the main conclusions from the trial were that at speeds up to 16 kilometres an hour, Bentleg openers were able to maintain low soil throw, 100% backfill and low draft requirements. But it also highlighted that there's a sensitivity to speed in Bentleg design. So where's the commercialisation at with Bentlegs at the moment? Well, the only work it's been done in a no-till sense is by a South African farmer called Danny Rossell, and he designed the RT blade with the aim to break up hard pans at speed, maintain 100% backfill and minimise surface soil disturbance. However, he used it in a split seeding system and only used it as a leading tine, and the disc was there to place the seed. So further research is required to understand the design parameters of the Bentleg and how they are sensitive to speed, and validate it down to a shallower soil so farmers in South Australia and Australia can use them. Also need to look at how we can place seed and granular fertiliser with it, as this has never been achieved in the field. My PhD is going to look at this, and it's going to use discrete element modelling to aid the design of it. Discrete element modelling creates a soil bin using lots of small soil particles and calculates the interaction between these as the tool passes through it. From there, I can select a grid similar to Dr. Ali Akbar's work and track how the soil moves with different openers. And from here, we can get the coordinates of the different soil traces and investigate different designs. I'll leave it there. Thanks.
Thanks for having me firstly. Today I'm talking about my PhD project, which is just over 12 months old. And I'm looking at the regional movement and sources of canola colonisation by diamondback moth in southern Australia. In my 10 minutes, I hope to give you a bit of a snapshot on what the project's doing, but also present some preliminary results from last year. The diamondback moth is the world's worst pest of brassica vegetable crops. It costs the world economy between $4 and $5 billion US annually in costs of direct yield losses and management. It's a highly migratory insect. It occurs globally wherever brassicaceous plants occur, and has a long history of evolving resistance to all insecticides used for its control. And currently, it has records of resistance to over 82 compounds globally. So in Australian canola, diamondback moth seasonally recolonises crops each season from alternative brassicaceous plants, which may be wild hosts, forage brassicas or brassica vegetables. Resistance is a major challenge for canola growers trying to manage outbreaks, as we can see from the photograph. Control failures during an outbreak can be quite devastating. At present, the origins of the colonising populations is not known, but we do need this information to start to develop some predictive models to forecast outbreaks and also design insecticide resistance management plans based on knowledge of gene flow between DBM populations from different hosts and different regions. To complicate matters, last year some Canadian researchers published a paper describing a new cryptic species of diamondback moth, which is now called Platella australiana. These were discovered in light trap samples collected from eastern Australia between 2004 and 2012. You can't tell them apart by looking at them, but they are quite genetically different and have differences in their reproductive structures. Because they weren't collected from plants, we still don't know what the host plants are and whether or not they're also a pest of canola. This is something we need to sort out quite clearly. The key question for my project is, what are the sources of diamondback moth populations that seasonally recolonise canola in southern Australia, and what are the implications for management? I have 5 aims related to that, which I will skip through in the interest of time, but they are in your paper. Essentially, studying large-scale population movement is quite logistically challenging, so I'm combining field-based monitoring, mainly focused around South Australia, but also some genetic techniques to understand gene flow. So, summer surveys, I want to know where they're over-summering. The black dots are where I did some surveys on Eyre Peninsula last summer. A regional network of light traps has been established in South Australia, and those are collecting daily flight data, so I'm looking for large-scale migrations. A regional network of sentinel fields was used last year to monitor the timing of first colonisation of crops by DBM. I'm using development and weather models to try and interpret some of that field data. And a large component of the study is population genetics, looking at gene flow between different regions and hosts. And thankfully there was quite a lot of DBM around last season, as many of you will know. And I had a lot of assistance from many of you, but also interstate colleagues in collecting a large amount of biological material for a thorough study this year. Amongst that, I'm also looking for the cryptic species by using a molecular assay. So at this point, I just want to pre-warn you that I am going to invite you to participate with me again in the research this season. So at the end of the talk, I'll put this up and you'll be able to text in if you are interested in working with me, which would be great. So just a little bit of data. I mentioned I did some over-summering surveys last March and April, which is actually autumn, if you think about it. I focused mainly on Western Eyre Peninsula because that is often where DBM problems arise first and are often most severe. And what I found was that Lincoln weed had a vast distribution right up that west coast. It was nice and flush in March. But another plant, sea rocket, which is a coastal succulent plant that grows along the sand dunes, you would never pick it as a brassica if you didn't know, but that also harboured quite a lot of diamondback moth. We did some resistance testing on several populations, and these over-summering moths had similar resistance profiles to populations from canola, which suggests that we do have potentially a refuge of resistant moths in that region over summer. And the second piece of data, in working out where DBM are originating, I wanted to know when they're first arriving in canola crops. So 40 agronomists from South Australia each adopted 1 sentinel canola field, or in some cases 2, and monitored those fields frequently from planting until first infested with diamondback moth. And then I used development models to back predict when the eggs were first laid in each crop. And if we go through week by week, the coloured circles highlight the weekly pheromone trap catch in each canola crop. So that's the number of moths caught in the trap. And the black triangles illustrate that the crop is being colonised by eggs or larvae. In other words, reproduction is occurring. So at the beginning of May, heading into the middle of May, quite a lot of canola crops were already being colonised by diamondback moth. And as we know, we had a big year with green peach aphid and beet western yellows virus because it was very warm and there was a lot of insect activity. So by early June, the majority of the sentinel fields had actually been colonised. So the upshot of that is that in 2014 there was widespread crop colonisation by diamondback moth in very early winter. Is this normal? I don't know, which is why I would like to do this again this year and hopefully under a more normal or average seasonal conditions. So in summary, the project is off to a good start, I think. I've really enjoyed and valued working with a lot of people in this room on generating some very unique data that will hopefully be of use to industry. The field work needs to be repeated to really start to understand the system, and really just watch this space is the message for now. But my final penultimate slide. Firstly, I really do want to thank the people who have generously contributed and chipped in and been part of a team working on diamondback moth with me. And I'll leave it with this slide here. Just would love to have the opportunity, if you are interested in monitoring a canola field or collecting a sample for inclusion in some genetic work, feel free to text in. This isn't locking you in at all. It just means I'll give you a phone call and you can decide from there whether you'd like to do it or not. And thanks very much for your time.